Hey guys, I'm going to walk through the making a protein simulation. So we're going to transcribe and translate a gene, which is some of our vocab words for transcription and translation. So I'm just going to kind of walk through the simulation with you to see, to get you to kind of know what you're doing. So you're going to choose a gene to begin, so you can click anywhere, and it's going to take you through transcription.
Inside the nucleus.
And it's going to have a little video for you. And then it's going to make you go through transcription and do it yourself. So we have our base pairs here. And we know that A pairs with U when we're making RNA. And then we have another A. We're going to pair it with U because we're making a strand of RNA. If we're making DNA, we would put a T there. But since we have an A and we're making RNA, we have a U. Now we have a T here. We know that T pairs with A, so we'll put that there. T does not pair with U. A only pairs with U. So we would not put a U here. We'd put an A here. G always pairs with C no matter what. Okay, and so I'm just kind of moving through there. Now we have A again. We know that pairs with U. You're going to walk through T for A. So you can get pretty good at this. So we just translated, or transcribed, sorry. We just transcribed a piece of RNA. We just did transcription. Now we're going to go through translation. So we have our strand of RNA down here. That's the instructions for making the protein. So our proteins do specific jobs for us. So this strand of RNA is like a set of instructions that needs to be read and decoded, so we can make a protein to do that specific job. So I'm going to show you how this works. So this is our ribosome. And then we have our strand of RNA, and we are going to try and find the correct set of 3 letters or anticodons to meet up with these 3 letters, which are called codons. So if I have AAG, I'm going to look for UUC because that would match those 3 letters. So I'd click on this one because it matches up. Okay, and then you can see here we have an amino acid that is going to be put together to make that protein. So these little green things are different amino acids which are going to eventually go together to make a protein. So if we're looking at CGG, we're going to look for GCC. So that is our gene up here. So that's going to come in. Then we have UGU, so we're going to look for ACA. It's just cysteine. GCU, we're going to look for CGA. CGA, we're going to look for GCU. And this is going to keep going until it is done reading the strand of RNA, reading the directions. So I just got done making that protein, so you're going to see how this is going to kind of fold up.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, aminoacyl tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
So you just transcribed and translated a gene. At the end of translation, we have a protein that folds up and is going to go out and do jobs for the body. So that's how the creating a protein simulation works.